So what I'm going to do this afternoon, two things. First of all, I'm going to talk about semantics. So we had heard the word semantics used yesterday by Martin, and I'll talk more about semantics now. So that's putting meaning on data. And then I'm going to show you some examples of database searches and what you can find, and also where some of the problems lie. But I thought I should start. Oops, what was that? No, yes, informatics is good for you. So it's not as bad as you think it is. Before I started, I didn't talk about. I was talking about data integration before lunch, and one additional database I should have mentioned I didn't get to was Ensemble. So a number of you know about Ensemble, and Ensemble is a combination of things. First of all, it's a representation of a model of the genomes. So they find gene models and they present them back to you. But they also integrate a lot of other data as well. So there are a lot of tracks in Ensemble. People find it quite difficult to use, but actually it's very useful if you know where to look for things. I'm not going to go through describing Ensemble. They have a lot of online help. But just to say that, for example, here, there's a gene model. And one of the things you can find are the phenotype data from MGI. So here's an example of database integration. MGI supply Ensemble with their or a representation of their annotations. And if you click on that, you'll find this part, for example, which is the MGI phenotype annotation, talks about the different alleles of the gene, and it tells you a bit of information about the phenotypes. And you can also get information about human phenotypes here. So the human ortholog of that gene, what kinds of phenotypes are associated with that. And as I say, we're about to talk about the semantics. And here, the phenotypes are very much the kind of things that someone with a clinical interest would write down. So, for example, growth retardation with deafness and mental retardation due to IGF-1 deficiency. Now, that is something which a human can recognize, but a computer can't. And so I'll come on to start talking about some of that. So, as I've just said, biological data is generally speaking recorded in natural language. As we heard this morning, natural language is what people write. So most of the biological data that's out in the world is in the form of publications, and that's in natural language. And natural language is something that's very difficult or effectively impossible in a really good sense for computers to interpret. Because it uses all sorts of symbolism, all sorts of rules to communicate knowledge. Now, it's the most expressive form of communication that we have, obviously, because we make language more sophisticated as time goes by. But there's a lot of ambiguity in it. There's a nice little example coming up just to show what kind of ambiguity there might be. And that means that it's very difficult just to take a piece of text, or it's impossible just to take a piece of text and do computing on it and get a sensible answer out at the end. Now, you might think, well, that's tough. That's tough. Computing people can't do this. But why is that a problem? Well, first of all, because we want to search that information. So, as I said this morning, if you want to search PubMed, you have to think quite carefully about what combination of terms to use. And you know very well that, particularly if you use the wrong terms, you get a lot of stuff back that you didn't want. And that's basically the problem of text searching. Data mining, which is a more sophisticated version of searching, it's a statistical field, but that's the art of extracting knowledge from large sets of data. Well, if you can't search data effectively using text, it's even more difficult to get knowledge out of that. And so if you want then to take text and transfer it between databases or put it into software, or compare between species, it's very difficult to use a human language. So we need to have a computing language that can represent that information in a way that computers can use. And here's an example of the ambiguity. So this is one example of ambiguity. So this is in OMIM. So OMIM, I told you this morning, is a database of clinical phenotypes, in a sense. And here's a set of text of terms used to describe, one particular phenotype, and they're basically all the same thing, right? And there's absolutely no, some of them are reasonably similar to others. Some of them are not at all similar to each other, but they're all basically trying to describe the same thing. And this is the problem of having people just write stuff down. Different people have different usages, or over time usages evolve, and the text just becomes difficult to interpret. And something else we want to do is link things which are expressed differently in different species. So this is the big issue, for example, of comparing phenotypes in mice to diseases in humans, and I'm going to go into this in a fair amount of detail. So there's lots of information about diseases in disease databases of different kinds. There's information about patients, or okay, we don't have access to information about patients, but we have information that might come out of patient records. We have information for clinical trials, and we would like to relate all that information to what's in the model organism databases, where some of the things are somewhat similar, although how you describe pathogenesis sounds rather difficult. Some of them are much less obvious. So, for example, phenotype and disease are not directly related concepts. I was talking about some of these disease databases. So, for example, there's a project called the Human Variome Project, who have just received quite a large sum of money, or have just negotiated quite a large sum of money from the Chinese government, actually, to build more databases of disease genes, individual disease genes. Now, but the problem with that whole group of people is that each of those disease gene databases is a unique database, and they all have somewhat different design rules, which means that actually bringing all that information together is going to be very difficult, even though they have money to make databases. And so we need to think about how we're going to do this kind of linking. So we have information in the literature which helps us to do that linking, but actually the big problem is how that information is represented. And it's worth remembering that what we're talking about here is information and not, or knowledge, not information, I should say. Knowledge, information, and data. So data is all the stuff that's out there. And so individual words in a text file are, in a sense, data. And this is the stuff that reaches our senses all the time in large amounts. But when we add meaning to the data, then that becomes information, and knowledge is what we extract from that information, the actual high-level meaning that we put into the structure that we put into that. And what we largely try to do in bioinformatics is to impart information, not data where it's similar, but information where we can. Knowledge is really something for scientists, I think it's fair to say, although, of course, it does get recorded in some sense. So let's look at this ambiguity question again. So there's a difference between the thing and what it's called, and this is something that we have to remember. So a lot of what we do in science and in biology, in science and in bioinformatics, is label things. So we have some sort of object, some sort of entity, we call it something. And what we call it is not what it is. And actually, psychologically, we have a concept in our minds of what this thing is, which more or less corresponds to that thing, and which is evoked by that label. And so a nice example of that is hedgehog. So hedgehog is a symbol, is a name for something. And we know it is a gene, or we know that the hedgehog family is a set of genes. And we know it is a protein structure. We also know it is an animal, which has nothing to do with the gene. And we also know it as the character, which is the origin of the gene name hedgehog. So if we have hedgehog in our system, we might evoke all sorts of different things depending on how that system is defined or designed. And we need to make sure that we only call back, say, the biological entities and not the cartoon characters. And that's the semantic definition. The semantic definition is what tells us what that symbol means in our domain of knowledge, and that's what we need to make, if you like. So I'm going to talk about ontologies, and some of you will, I guess many of you will have heard of the gene ontology. How many of you have heard of the gene ontology? Go on, some of you must have heard of it. I know you have. So the gene ontology is the classical ontology, but I'm going to sort of generalize it to start with. Then I'm going to come more specifically into how that's used in the phenotype domain. So the first known ontology, and again I owe this to Paul Schofield, was put together by Isidore of Seville in 600 AD, so that's 1400 years ago. And his idea was that he would try to encapsulate all knowledge, which was something that in those days they thought was possible, into a structure. So a framework for all knowledge, which is constructed hierarchically, and that's still what we basically do. And this will be useful for labeling. It wasn't designed for computer science because it didn't have computers in those days. It wasn't designed for helping people to think about knowledge. It was just a way of codifying knowledge, but it was the first example of this concept. And so in the modern world, there are at least two uses of the word ontology. One is in philosophy, and this is, in a sense, a representation of what we know. And one is in computer science, which is really a way of structuring concepts. And they're almost the same thing, but not exactly the same thing. And what I'm really going to talk about is the computer science use, although it's very similar to the philosophical use.
But where does Barry Smith enter into this game?
Say it again?
Can you tell us along the way where Barry Smith enters into this game? At which level? Okay?
This is the philosophy versus computer science. I think. So Barry Smith is the head of the National Center for Biological Ontologies in the USA. He's a philosopher by training. And there's an interesting history of biological ontologies, which started out with the gene ontology, which was originally created by Michael Ashburn in the UK and people in the US. Along the way, Barry Smith decided that this was something that he really wanted to get involved in. And he started to criticize what was done in biological ontologies on philosophical bases. And so this has resulted in the restructuring of the ontology, and along philosophical lines, which a lot of biologists don't think was really a good idea. But nevertheless, it has happened. So what he's tried to do, I think, is introduce some philosophical rigor into some of the structures that we use, or at least the gene ontology and people like that use. So you can a lot of these definitions sound quite vague, but they do mean something. So an ontology, it's not working. Can be defined as a formal conceptual representation of a domain of knowledge. So that's both information and the relationships between those bits of information. And really, the idea is that if people can agree on a structure like this, a conceptualization like this, then they're essentially all agreeing on the nature of that knowledge and that domain of knowledge, and so they can then work together by using shared terms and shared relationships. And so you need a set of names, and it has to be accurate, and it has to allow logical inference from the structure. So you can make, just as a simple example, here's a simpler example of a bit of an ontology. So under phenotypes, there is sexual phenotype. There are three, I think, possible sexual phenotypes, male, female, or intersex. This should be complete in the sense that it should describe all possible outcomes. This isn't always going to be the case in ontologies because it's they rely on knowledge, and sometimes not. Well, knowledge is generally incomplete, and therefore they will expand and grow. But in principle, an ontology should have all the knowledge in a domain in it. So the components of an ontology are, first of all, the concepts, so the things that we're thinking about when we talk about a particular domain of knowledge. So it might be a protein or a gene or a process or whatever. And it's important to keep those different things apart. So you don't want to have an ontology which has processes and genes in it. You want to be able to have those distinct groups of concepts. So those are the sort of, that's the concept level. You need to have names for the individual things in there. So sonic hedgehog or hexakinase. And you need to have relationships. Now, typically in biological ontologies, there are two kinds of relationship. One is the is a relationship. So glucokinase is the hexakinase. Or they could be part of. So you can have eye as part of the head, for example. So those are the classical relationships in biological ontologies. And as I said, the point of having these things is that you are encapsulating the knowledge of a particular field, and that allows you to communicate with other people because everybody should understand what you're talking about. There's an element of standardization in the sense that you have a word for something. Now, that's an opportunity and also a problem because not everybody wants to use the same word for the same thing. But what ontologies typically have in them are synonyms so that they can be translated. So you have a thing and you have more than one label attached to it, or you will have one standard label. That allows you to do that without too much unfortunate argument. And it should allow you to do computation. So, for example, you should be able to match something in one species to something else in another species because they both belong to the same concept. They have the same concept even though they're in different species. You can do hierarchical searching. You can do searching and recovery of data. And potentially it also helps you with statistical analysis. And actually, at the end of the day, after I've gone through a little database thing, I'm going to show you some of the work we've been doing, which is almost systems biology. And that actually makes use of ontologies to try and relate phenotypes to genes and diseases. And so that I can show you a little bit of an example about how that's relevant to statistical analysis. So what do we have? So that's the sort of general idea of what an ontology is. And you may have noticed, and I haven't said anything about it, that the abbreviation, the undefined abbreviation CV comes up occasionally. And CV is a controlled vocabulary. A controlled vocabulary is almost an ontology, but not quite. So a controlled vocabulary is a list of terms that you would use to define, to describe an area of knowledge. It's not the same as an ontology because it doesn't contain relationships. So it has terms but not relationships. And many of the things that we have in biology are actually controlled vocabularies in the sense that they are a set of terms that people can use, but they're not ontologies. So this is, we've said a few times today already that human phenotype is a disaster area, and this is sort of why. So these are some of the controlled vocabularies used to describe human disease. And this is so one level of the disaster area. So we have Galen, which is actually, I think, an ontology, which is designed for human clinical application, but it is very complex and it's licensed, which means that not everybody can use it, which is a bad thing as far as we're concerned. We have ICD-9, and in fact there's a follow-up, ICD-10, which is the International Classification of Disease, and that's designed for human epidemiology, and it has weaknesses if you're strict about how you design your ontologies, because it isn't just names. The names imply processes, and that means it's actually got two different domains of knowledge within it, and there are other issues as well. SNOMED, which is widely used, and it's accompanying SNOVET, is a structure which is basically designed for medical billing. So it has lots of concepts in it which are not diseases. It's like, fell off his bike. So he fell off his bike and broke his leg, because that's an insurance term, sort of. So there are things in there which are not diseases, which makes life more difficult. Then we saw earlier MESH, which is fairly simple, a list of terms. That's a toxicology, control vocabulary. This is more cancer-related toxicological pathology method structure. And UMLS, again, which we saw earlier, very large and complex and not really an ontology. And so one of the issues that we often find is that people are satisfied to come up with a list of words, and what they don't really think about is how those words are related to each other, or they use a very simple conceptualization of that, and I'll probably come back to that later. And most of the ontologies, possibly all of the ontologies that we use in bioinformatics, have put more emphasis on what's in there than how those things are related to each other. And for bioinformaticians, actually, it's very important to know about how these things are related to each other. And sometimes people don't really think about what they're trying to do when they build ontologies. So that's the human one part of the chaos in human description, disease description. There are more ontological descriptions. So there's a couple. There are four ontologies listed here. Two of them are human. So there's something called the disease ontology, which ought to be what we all want. And there's also something called the human phenotype ontology, and I'll tell you a bit about this in a second. So the disease ontology is an ontology of diseases, but it, as I understand it, has suffered from a number of problems, and it may actually not be being maintained anymore. The human phenotype ontology is derived from OMIM, so it relates quite nicely to OMIM, which is a database that people are interested in using. But it has its own problems because it's been generated automatically, hasn't been curated until it's starting to be curated now. But it wasn't curated for a long time, or for some time.
Who is this guy from?
Peter Robinson from...
this guy who is in Cambridge.
So HPO is Peter Robinson in Berlin. George is doing Pato, which is different. And then we have, in the mouse, we have the mammalian phenotype ontology, which is the ontology developed at the Jackson Lab to describe mouse phenotypes, which is actually pretty good. And Paul Schofield's ontology of pathology, which is also pretty good. So we're in a position where we have better ontologies in the mouse than we have in the human, which is good for mouse people, but not so good for trying to do things with diseases. If you want to look for these things, there are a couple of services. So there's at the EBI, there's an ontology lookup page, which allows you to find ontologies. You probably won't want to do this, but nevertheless. And there's also Obo Foundry, which is where people who are developing ontologies lodge their latest version. So if you want to know what the latest version of a particular ontology is, including the gene ontology, you'd go to obofoundry.org, and that's where you would find the latest version. So I don't really want to go into massive amount of details about individual ontologies, but I suspect you're not really interested in the details. You can have. I mean, these slides are obviously in the reference set, so you can look at them. One problem. This is the disease ontology. One problem with the disease ontology is that it's been a bit inconsistent. So it started off using ICD-9. Then it moved to other terms, so it starts to incorporate different terms from different control vocabularies. And then it gets reorganized. And so people, A, find it very difficult to follow, and B, it's likely to have terms for the same thing in different parts of the ontology, which is very bad news for anybody trying to do anything. The human phenotype ontology, as I said, was derived from OMIM, and what happened was that this guy, Peter Robinson in Berlin, decided that he really needed to have an ontology which just described those bits of phenotype, human phenotype, which are in the clinical synopses of OMIM. So OMIM has a page for each disease called the clinical synopsis, and what this describes is the phenotypes which are seen, which are described in the full disease page. And so it covers Mendelian disease, and any term that's been in that massive great list of phenotypes, which appears more than once, is put into this ontology, so it's something that can be used. And so there are something like, there's probably more than that now, but something like 9,000 terms in the ontology. The weakness is that because it's automatically generated, it's not particularly well structured. Things are put together just because they sound similar, not because they necessarily are, semantically related. So there are issues with it. But it's the best thing we have actually in the human domain, because it gives us a set of phenotypes, many of which are quite similar to mouse phenotypes, that we can use to link. And I'm going to tell you a little bit about that linking process in a minute. So this is a key, I think, entity in the description of human disease. And then I'm just going to come on and talk about the mouse ontologies. So mammalian phenotype ontology, as I've said, is a very useful key tool. And when we come to look at some of the database searching, you'll see that it's quite often used. So it's not just used at the Jackson Lab, it's also used by other databases as a way of getting into mouse phenotype. So it's a very commonly used structure. And although you can't read this, obviously, basically what you have is a high-level structure of about 30 terms, which are things like neurological phenotype. I'll pick a couple of these out. Lethality, pigmentation phenotype, reproductive system phenotype. So the top level, if you like, of this is very, very broad concepts. And then below that you'll have much more defined concepts which allow you to break down the phenotype in more detail. So because it's derived, so this has been developed alongside this process at MGI of building up their database. So it's basically the set of terms that they use to describe phenotypes. And as a consequence, it really pretty well has terms for everything that's in the literature, which is, very useful. In other words, it's what we call granular, in the sense it has a lot of, you can go down and define just about anything in phenotype that's been published using this, with some exceptions. But generally that's true. It's sort of, it is a proper ontology. It, to some extent, supports inference. So, for example, I think there's one here. Well, so things about tumours will be classified under tumorigenesis. So you can infer that that's about tumorigenesis. So that's useful. And of course, it's useful for literature annotation because that's what it's been designed for. So it's pretty useful for many things. The pathology ontology, which was developed by Paul Schofield in Cambridge, is specifically that. So it's an ontology for mouse pathology, although they're currently trying to expand this into a general pathology ontology, so it will be used for humans and mice, because actually there's not an awful lot of difference generally between humans and mice in that area. So this was developed by a set of pathologists. And in fact, I think it's true to say that Paul is neither a pathologist nor a bioinformatician, so that's a good start. But he has got pathology friends, so they've done a reasonably good job. There are some differences between human and mouse, and particularly when in transgenic animals, genetically modified organisms, sometimes you get peculiar outcomes. There is an issue which is a common issue in ontologies, which is that bioinformaticians want one thing from them, and the biologists working with them want something different. So in terms of structure, for example, the organisation of empath is one that would be familiar to pathologists, which is in many ways good, but there might be issues with that for other purposes. Yeah. And it doesn't have some quantitative features, but that's not surprising. And so one reason for introducing that, apart from the fact that it's a very good ontology, is that it's useful. It allows us to introduce a concept which is the combinatorial approach to annotation. So this is just a diagram of a part of, there's a mouse anatomy ontology. This is another ontology which describes mouse anatomy. And you can see that one of the features of ontology generally is that there are various routes to the top. Now, this is one of the areas where Barry Smith has come into the fray, because Barry Smith doesn't believe that there should be multiple routes to the top, but actually in biology,
there shouldn't be
multiple ways of getting to the top of the ontology. But in fact, in biology, we often have different ways of relating things. So, for example, gut epithelium here is part of the gut, and it's also a form of epithelium. So the I there is a relationship. The P is a part of relationship. So that concept there is part of two different systems, and both of them make sense, and both of them are necessary for biologists. But it's somehow regarded as sort of heretical to do this, but it's actually.
What are these numbers?
So the numbers are, very good question. I think they're levels. They're probably IDs. So going down from the top. So usually the top level of ontology is labeled 0. Then as you go down, you get more fine-grained levels. Although that doesn't work here, so I'm not quite sure about that.
Yeah, but the numbers, it's not that
Yeah.
at the top they're small.
No.
So
Yeah, I don't know what they are. I don't know. This is not my slide. So this is the mouse anatomy ontology, and the point about that is that you can. So one of the problems with something like pathology is that you can have the same pathology in many tissues. And so, in theory, if you described the same pathology in every tissue, you could have a vast ontology, which would be very repetitive. And this is not the ideal situation. And so, this is a concept called the cross product has been introduced. A cross product is basically using two ontology terms to describe something else, and I'm going to use this more later on. So here, for example, you can have examples. But anyway, the idea is that you would have a particular pathology associated with a particular piece of anatomy, and this would be something which would potentially be part of a bigger ontology, but which you don't really want to make. And this is a useful concept for phenotyping as well, because in phenotyping you have you want to be able to associate values with things. So I'll tell you about that in a second. So remember this concept of the cross product if I use it again. So there's some concepts I've introduced. How can we use this, what we have? Our problem is, actually, this is the other level of chaos. Our problem is that diseases are not phenotypes. Diseases, as we heard from Wolfgang this morning, are abstractions of something that's wrong with the organism, which are characterized by a set of phenotypic observations of some kind. And so, well, one way of looking at this is that diseases are composed of disorders, which along with severity and progressivity define the disease. From an informatics point of view, if we want to relate phenotypes in mice to diseases in humans, we need to know what it is about those diseases in humans that define them in a way that we can use that phenotype information for. So there's some examples here. So this is, again, we heard something about, again from Wolfgang, about different diseases having overlapping phenotypic characteristics. This is another example of that. So 3 diseases which have many common features. So these are, Sotos-Weaver syndrome, Beckwith-Wiedemann syndrome, Simpson-Galabi-Baimel. And they have unique features, but they also have shared features. So the ones in red are common to all of them. And so if you only have information about the red ones, you're never going to be able to distinguish these. And if you don't know what the other ones are, and which ones are unique and which ones are shared between which ones, you're never going to be able to relate phenotype information to disease information. And so the issue this actually raises is, how do we know, how does an informatician know, or how does a mouse biologist know which phenotypes are relevant to their diseases of interest? Or if I have some phenotypes, if I have a characterization of a mouse, how do I know whether those phenotypes are relevant to a particular disease or which disease they're most relevant to? And these are the kind of questions that we are starting to have to answer in projects like IMPC. So IMPC is going to produce a phenoset for every gene knockout. How will we know which diseases those are most relevant to? Who's going to work that out? If there are 20,000 pieces of data or data sets, is somebody who's interested in some obscure disease going to trawl their way through all 20,000 of those to find something of interest? Maybe not. We need to have tools which will help them to find what they want to find.
So which of these proportional mathematical function, you would call them phenotypes?
So
how do you call these categories?
Yeah, that's a good question. I wouldn't necessarily call them phenotypes. They're I guess well, I wouldn't even call them diseases. I wouldn't even call them disorders, I think. I don't know what you call them, to be honest. I mean, clearly that, for example, macrocephaly is a phenotype. I think that's fair enough.
They are not all phenotypes.
But they may not all be phenotypes. And that's, so the big issue, one of the big issues is going to be defining diseases in terms of phenotypes. And this doesn't solve that. But it does point to that.
And then, this is just, it's truth conditions.
Yeah.
You see, that in this part in which there is overlap, all these things are true in both.
Well, I think it's worse than that. They may be true in both. So this can be a characteristic of that and of that, but they're not necessarily essential. So that makes it even worse, right? So we need to know that too. And some of these are essential, some of these are not essential. It's complicated. Right? So
this is a mess. Right?
It's very, very difficult. Well, so it's a result of the natural way that disease description has grown up, because it's a human process, and people describe things in a particular way. The issue, I think, is that what we need to do more sophisticated analysis is to break diseases, or to describe diseases in terms of phenotypes. Who's going to do that? So this comes back to the political issues, almost more than the technical issues. So people have got to sit down and do this, and that's going to be hard to organise. So here's an example. So this is HELP syndrome, and which is made up of a number of different phenotypes, which are reasonably actually good phenotypes. Low platelet count, elevated liver enzymes, a bit vague, but anyway. Hemolysis, which is clear. So this is, if you like, a disease, and it's actually one that's quite well defined in terms of phenotypes. And this is really just to show where the problems arise. So if you search MGI for HELP syndrome, so you get a number of genes back, sorry, not just 3. But what you get is 2 genes here which are in mouse show the phenotypes of the disease, but which are not associated with the disease in humans. And 4 genes which are associated with the disease in humans which do not show the phenotype in mice. So what do we do about that? What I would say most likely it's reasonable to say is that these genes in mice which don't, which show the phenotype but which are not found in human, might well be part of the same pathway as the genes in human, but because of background genetics, they don't show up in human. But so what this is an attempt to pick out the phenotypes of HELP syndrome in both species. And I'm sorry this is quite a messy and complicated slide, but I'll just try and point out the main points. So basically, all of these terms are part of HELP syndrome, which is the human disease, if you like, on the right-hand side. On the left-hand side, what Paul's done is put down the mammalian phenotype terms, which may or may not match. And you can see that some of them are the same. So hypertension is a mammalian phenotype. Many of these actually fit quite well. That's, okay, not the same, but you can see, we can see that they're the same. But a computer can't see that's the same because they're not the same words. And so this brings in the second column here. So this second column is what we call logical definitions. So a logical definition is a breakdown of the original term into smaller atoms out of ontologies, which are applicable across species. That's really the main aim. So in this case, for example, hepatic failure, liver failure, they're different terms, but they're both in Go hepatobiliary system processes, and they're both dysfunctional. So they're both that in both species, and so you can, by knowing that they're the same thing, that these things have the same breakdown in the different species, you can map them even though the terms are not the same. So when I say you, I mean a computer can map them. Again, glomerular vascular disorder, which is not exactly the same as abnormal glomeruli, but that's the best we have in MP, in mammalian phenotype. But they can be mapped across because they are abnormal glomerulus concepts. So again, if you break these kind of things down, you can map across species even though you don't have an exact match. Yeah, and this one is particularly complex because I'm not going to show you don't think I'm going to show you the breakdown of this. But because this is made up of actually two subconcepts, and it's even more difficult to do. But it is possible. There are methods for doing this. So what I'm just going to now describe is how we do these breakdowns a bit. What's the basis of the breakdown? So the mammalian phenotype ontology is what is known as a precomposed ontology. And that means that all of the terms in there can potentially be broken down into smaller parts, but it's been built this way because these are terms that people recognize. So there's some examples. So positive or negative regulation of mitosis is regulation of mitosis with a value which is positive or negative. Increase or decrease angiogenesis is angiogenesis which has a value of increase or decrease. So you can see how you can break these down into more sophisticated subsets of concepts. We don't normally do this because actually what people want is a simple term they can use. So when people are annotating mice, they don't want to build up these complex concepts, and so we have these precomposed ontologies. But it's problematic because these things can increase, you can have increase and decrease of everything. So why would you have it in that bit of the ontology, for example? So there's a tendency for these ontologies to get bigger and bigger and bigger without actually adding real knowledge to them. So the alternative is what we call post-composition, which basically is what I showed you both for the pathology and for that disease breakdown just now, where you take a number of different ontological terms and fix them together. And by doing that, you can replace part of that from different species and map them together, for example. So going back to that, what you would have would be that Go term, angiogenesis, and you would have some value, increased or decreased. And those would be the two bits that would describe this concept. So this is logically rigorous, and it allows us to describe things that we can't describe with things like the MP. So we can describe quantitative data, for example. We can describe the value of something. Rather than just saying it's high or abnormal, we can say it's got a value of 0.3 or something like that. So that's good. And it also allows us to do more complex logic on the structures. It does, however, make the statements very complex, and it's not the sort of thing that the individual person annotating their mouse would really want to use. So what we really need to do is have a bit of both in our informatics system. And I should say that George Goutos, who works with Paul in Cambridge, was instrumental in developing this approach to phenotype description. So what we have worked on for some time is basically combining these core ontologies like anatomy or behavior, if we had a behavior ontology, or pathology, with an ontology called PATO. PATO is the phenotype and trait ontology, but actually it's an ontology of what is unknown as qualities. So qualities being bigger, smaller, wider, yellow, these things. And that allows us to build up phenotype descriptions. And we've actually been experimenting with using this in the Europhenome database. So the idea is that for that mouse, which I don't think you can see actually at this resolution, but anyway, it's got sparse coat hair. And so what you do is you combine something in the mouse anatomy, which is a trait. Sorry, it's not a trait. It's something in the mouse anatomy, mouse coat hair, with either a concept which is distribution, and by combining coat hair and distribution, you get a trait. So coat hair distribution is a trait. But under distribution, you have various qualities of distribution. So you have sparse or dense or various things. And so if you pick that, you have a phenotype. So you migrate from having a trait to a phenotype by adding lower levels.
Level properties.
Yeah. So the thing about PATO, which is sort of is unique, is that you migrate from descriptive properties to general properties, and by doing that, you migrate from trait to phenotype, which is unique. Which means you only need two terms, but implicitly in that is that as well. So we've been experimenting with using that in Europhenome with the aim that eventually it will be possible to map between the mouse phenotypes and the human diseases, although we're still quite a long way from doing that. So that, as I say, the phenotype and trait ontology, that's PATO, is a core ontology, so that means it's in that the oboe foundry is a recognized good quality ontology. And it's partly designed for capturing data on individual traits. And not everything in it is actually phenotypic, but nevertheless it will do that job. And so this is sort of what I was saying about between species. So basically, if you have, maybe I should just pick one of these. So you have eye color, which is a trait. Now this part of eye color is eye, but that could come from any species. So you can have Drosophila eye, mouse eye, human eye. And if you have externally the links between eyes in different species, immediately you can link the eye color trait in Drosophila to the eye color trait in humans or mice computationally. And so that's really the objective of doing this. One thing you need is a way of linking between species. And this is still, well, it's in development, it's coming along. There are two approaches that are out there at the moment. One is Uberon, which is a multi-species link between anatomies, which has been generated largely automatically. And the other one is something I'm involved in, which is called the vertebrate bridging ontology, which is going to be manually constructed. So, because linking anatomical terms just using text mining is not always going to work. So you need to have a bit of manual input. And so we're trying to build the VBO at the moment just to link between vertebrates, so that we know that this bit of a fish is homologous to this bit of a mouse, for example. And there are a number of other ontologies which relate to this, to these. Well, I think to Uberon particularly. So there's you can start to combine ontologies, and once we start to bridge ontologies, then we can start to bridge phenotypes. And when we start to bridge phenotypes, at least we get rid of one of our problems, which is transferring across species. We still haven't got rid of the problem of disease description, which is a bigger problem we have to deal with. So, so these are the bridging entities. So we have blood, which is common to everything in mammals anyway. We have liver. We have a necrotic process, which is common. We have renal glomerulus, which is common. So we have in that bridging ontology, we have shared concepts which we can use to link across species. And that's the tricky part. So this is where we have problems. So these are the two parts of hemolytic anemia. There are lots of terms, and some of them map well, and some of them don't map well. And there's spherocytosis in NP, which doesn't map across well. So there are bits of even a very simple disease description which don't map well, and that's all has to go into the mix somewhere. And I'm coming to the end of the ontology stuff now. Hopefully you've got an impression of where the complexity lies. It's about description, but it's about making sure that descriptions are rigorous, and that's really the hard part of doing it. These are just a couple of tools that have been built to help do this cross-species linking. There's a database called OBD, which is run by the gene ontology people, and a piece of software called Rosetta, which, again, you see that this is a bit of manual, a bit of automatic, and if the right people aren't doing things in the right way, those links may not be right, and therefore the whole thing will fall down. So every step in this process is quite critical in terms of eventually linking between, say, mouse and human. Lots of people helped out with this. Paul has run the Empath project, and these are people who have helped him, and these people too. Susie Lewis, Chris Mungle are part of the Go Ontology group. Peter Robinson developed the human phenotype ontology. Michael and George work with Paul. So that's the end of ontologies for today.
Thank you.